Hello, welcome back, everyone. It is my great pleasure to introduce our final keynote speaker of the conference, Clay Reid. Clay is a senior investigator at the Allen Institute for Brain Science, where he started a department in 2012 to study how information is encoded and processed in neural networks of the visual system. Prior to joining the Allen, Reid was a professor of neurobiology at Harvard Medical School and has had a long and distinguished career as a visual physiologist, using both electrical and optical methods to understand the function of the visual system. Around 2005, he took a brave leap away from physiology, or in addition to physiology, and was an early adopter in the renaissance of using large-scale electron microscopy techniques in order to map the detailed networks underlying the physiology he spent so long studying. So today he'll talk to us about some of those efforts in his talk entitled Large-Scale Circuit Reconstruction in the Cerebral Cortex.
Thank you, Forrest, and thank you to the organizers for inviting me. I originally planned to give an introduction and show a lot of current data, but it's the end of the conference. You've actually heard some fantastic work in the topic I was going to dive into, EM connectomics. And I will talk about EM connectomics, but also another kind of connectomics with light microscopy in the human. So I thought I would make the talk more about how we got here, where we are, and what might be the exciting big projects and big goals for the next decade. So the title is Large-Scale Circuit Reconstruction in the Cerebral Cortex, but it perhaps should be that plus prospects for whole brain mapping with light and electron microscopy. So standing way back from, I guess, how I got into the field of anatomy, large-scale anatomy, large-scale EM connectomics, I've always been interested in the relationship between what neurons do and how they do it. And much, if not all of that work, has been in the visual system, relating the function of individual neurons and the connections, the connections that might underlie the computations. And people always talk about the computer analogies for the brain, but there's one that I kind of like, just for the visuals alone. Way back when, the visual for a computer was as often as not a picture like this of a computer. I'm missing the image right here, which helps. There we go. I touched it. The instrument panel, the light panel to show where you might be in a calculation. And this is the panel from a reconstruction of arguably the very first programmable electronic computer, Colossus, which was used at Bletchley Park by Alan Turing and others to crack the Enigma codes during World War II. And the pithy line is, Colossus helped to reverse engineer Enigma. How might one reverse engineer Colossus? One idea is to look at the instrument panel and say, these yellow lights look somehow like a base 10 counter, or two banks of 5 adding up to base 10, but wouldn't it be great to have the wiring diagram? And I went into YouTube and found this, and I went into the internet and found a white paper on Colossus, and here is the biquinary, the decimal counter that's in Colossus. And the reason I did this is that an instrument panel always reminded me of what calcium imaging looks like. In 2001 or something, my group switched from electrophysiology to using 2-photon imaging to image calcium in neurons, which these calcium transients are a very good readout of spiking activity of neurons. And then the reason I switched, or we switched, from electrical recordings to optical recordings is you can do many neurons at once, but also it's an essentially anatomical technique. And wouldn't it be great if you can use this anatomical technique to somehow bootstrap your way into a secondary anatomical technique that can give you connectivity? And electron microscopy was sort of a vague idea that one might be able to do this. But I think I have, the next slide, I just found this a few months ago. A surprising number of neuroscientists of almost exactly my vintage were inspired from 1979 to 1980 something to study the brain by seeing this amazing issue of Scientific American that had a series of articles about all aspects of brain science. There's an article by Hubel and Wiesel which really changed my life. Hubel and Wiesel, who did all the pioneering studies of visual cortex, and reading that just inspired me. But I also read several other articles, including, this is in 1982, I think, this very high-level article by Francis Crick, which, in retrospect, really presaged a lot of the techniques that have become the go-to techniques in neuroscience, from up to genetics to many other things. But I had forgotten, but maybe had internalized this extraordinary sentence. This was in the middle of a paragraph about all the great techniques that one might do, but we still need theory. We still need theory in order to figure out what experiments to do, because we can't do everything. And the sentence is, it is no use asking the impossible, such as, say, the exact wiring diagram of a cubic millimeter of brain tissue and the way all of the neurons are firing. And this actually has been sort of the holy grail of my career, but of the recent history of connectomics. And I think this is actually where we are, with just amazing technology, from different types of microscopy through high throughput data collection, through the extraordinary type of analyses you've heard about all week. This is now possible. So that was, whenever it was, 40 years ago. Wouldn't it be amazing if really the starting gun for the renaissance of electron microscopy that Forrest spoke about was this fantastic paper from Winfried Denk's group. Winfried Denk, who pioneered two-photon microscopy, which allowed all the functional imaging. And the idea was, why, in this modern era with high throughput data collection and just instrumentation, why can't we take three-dimensional electron microscopy, serial electron microscopy, into the 21st century? And I won't talk about the exact techniques, but the exciting thing was the idea, and for me, the most exciting thing was going into the journal website and downloading a whole bunch of supplementary data sets and stitching them together to see this for the first time, which is 40 by 40 microns in X and Y of electron microscopy of the cortex, and stepping through in Z, to make it three-dimensional with serial sections. And if you're not used to seeing EMs every day, especially this sort of grainy rendition, it's very hard to see what's going on. But optic flow is an easy thing. If you see things moving like that, that's a large neural profile, in this case a dendrite, that's sort of oblique in 3 space as you're cutting through it. The circles obviously are the nuclei that are sectioned, and now it's the one at the bottom is the middle, and now that's the very end of the roughly spherical nucleus. So this was the inspiration really for all of us. Let's do this. And as Forrest said, our group started in 2005. Not too long after this paper. Let's do this. Why do it? And there, as you saw, our group and others have great interest in relating structure and function of cortical circuits, but there are just a lot of basic questions that one can answer with these extraordinary data sets and the recently extraordinary segmentation of the data sets. Simply morphology. If you have a data set that densely captures the data for a volume of any biological structure, but certainly for neurons, you have an unbiased sampling of all the neurons there. What are the morphological classifications? Cell types is one of the fundamental principles in neuroscience. There are many cell types. What are they? How are they connected? What do they do? Connectomics, one can ask what are the determinants of connectivity. Topographic specificity, these are, in the next slide I'll say a bit more about these 3 categories. Topographic specificity is neurons are not going to connect unless the output, or the input, the axon, connects to the target, the dendrite, overlaps. They can't connect unless they're in the same place. Cell type specificity, where you are does not determine completely who you connect to. There are very strong rules, and increasingly interesting as the more we look at it, strong rules that certain cell types with high probability and high density connect to other cell types, but not to a third cell type. And then finally, functional specificity. How does the function of the circuit affect or influence whether neurons are connected, and obviously vice versa? How's the connectivity lead to function? And then there are a whole bunch of things that perhaps are more nerdy for neuroscientists, but are absolutely crucial. Morphological correlates of synaptic weight. How can we look at these morphological things and say, with high probability, I believe that is a strong synapse that, from other literature, says it injects 3 nanoamps of current, et cetera, et cetera. Synapse location, how that might affect the integration of many inputs to a cell, et cetera, et cetera. That's the end of my introduction to microscale connectomics. But I thought I'd talk about something else, a new project, a very new project for our group, and really, I think, for the world, which is the prospect of doing dense wiring diagrams at a slightly lower scale, but it's still at a very kind of extraordinary scale of the human brain. And this I certainly remember being inspired again by Francis Crick, just an extraordinary paper slightly more recently in 1993 called The Backwardness of Human Neuroanatomy, where he and one of the great anatomists, Ted Jones, wrote specifically about human neuroanatomy. We want to know who we are. We want to understand the human brain. And there's been a great we've heard this week of great advances in human neuroanatomy, molecular neuroanatomy, et cetera. But very specifically for the wiring and the large-scale wiring of human brains, there's a backwardness. Even today, whenever it is now, 25 years after this paper, that the great advances in the wiring of the brains of experimental animals has not been applied to the human, largely because the techniques that you can do in experimental animals are not one can't do these in human brains. And early in the paper, he says, clearly what is needed for a modern human brain anatomy is the introduction of some radically new techniques, ... And he uses an example in the paper, a fairly recent back then example that many people might have seen. It's one of the famous iconic figures from the last 25, 40 years. This is a figure from Fellman and Van Essen of the interconnections of the 20 plus or minus, mostly cortical areas in the macaque brain that are responsible for vision. And there is a very nice wiring diagram that he made for this. It's a similar wiring diagram is hot off the presses actually in the visual system of the mouse from the Allen Institute. And there's a whole bunch since that time of extraordinary work of large-scale wiring rules, area-to-area rules from a great deal of work done here in the mouse and elsewhere in primates. But he asked, what does the human equivalent of the connectional map of figure 2, that's figure 2, look like? And in straightforward, non-scientific prose, the shameful answer is that we do not have such detailed maps because, for obvious reasons, the methods that we do in animals can't be applied in human. I think that has changed and changed quite recently, not entirely because of new techniques, but because of the ability to collect very large data sets. And this is just the introduction. I'll talk about it at the end of the talk. What's the current state of whole brain connectivity? It's from diffusion imaging of human brains, which has been extraordinary. I'll talk about it later. Has been extraordinary for sort of setting the large-scale picture of things, and in particular, as it relates to functional imaging, it's really a fantastic science of human large-scale, very large-scale connectivity and function. But why is this not really a wiring diagram? And the answer is that the voxels are on the order of a millimeter, maybe half a millimeter now. And each voxel has a mean direction, and maybe sometimes a mean direction and it's orthogonal, but a mean direction. And the wires come from linking together those vectors. They're emphatically not single wires. But with antibody staining, some clearing of the tissue or expansion of the tissue, and things like light sheet microscopy, it's possible actually to look at individual axons, sort of pick your resolution, and the resolution gives you the size of the axons that you can discern. But remarkably, to me, we've been doing this for a while, and the individual axons are very traceable. I'll talk about that later at the end of the talk. And the details in this little blurb in the right corner I'll get to at the end of the talk. So, and from that, what might we learn? Sort of a bunch of fundamental questions. What are the areas defined by projections? Are these areas that we define from projections the same as the coarse architecture that defined by other methods? I'll get to the questions at the end, and I'm sort of giving too much introduction, but there are a lot of fundamental questions that one can get from this uniquely detailed map of human neuroanatomy. Why is all this possible? All of this is possible now essentially because of the big data revolution from data acquisition, storage, and analysis that we've been talking about all week. But one fundamental question is, how big are the data sets that one needs to just get in the door to do this kind of science? And up in the upper left-hand corner, I'm going through a powers of 10 romp from the synapse scale to whole brain scale, and asking how much data you need just to do these studies. And the figures of merit in terms of how big the data sets might be, is that for EM, you need on the order of 10 nanometers cubic voxel. voxels. And for light microscopy of these wires, of these long-distance axons, you need on the order of 100, 200 nanometer, 10 times larger voxels. But with EM-size voxels, there's 1 micron. Fun fact, there's 1 synapse per cubic micron. But just to see 1 synapse, you need a megabyte of data. A cubic micron is a megabyte of data. Just 10 nanometers goes into 1 micron. 100 times 100 cubed is a megabyte, or a million, a megabyte. Can't do much connectomics there. Let's say, ask, what do you get if you have if you have 10 microns, 10 times bigger? 10 times bigger is 1,000 times bigger in data. And essentially, you get 1 cell body, or most of the 10 micron, more than 95% of the 10 micron cubes in the brain have no cell bodies. But it's the size of a cell body. Again, you're not going to get connectomics. How does neuron A connect to neuron B? To get in the game, you need 100 microns. And here's from data you'll see later on. This is a neuron in the cortex whose nearby arbor, whose proximal arbor of its dendrites, it's fairly cut off, but you can see a lot of stuff going on within 100 microns. You can see a small fraction of its axonal arbor, but you can get in the game. You could imagine finding the connectivity between some neurons at 100 microns. But that's why it was so hard to get this field started 15 years ago, a decade ago. You need terabytes and terabytes of data to really look at networks. It's certainly in the cortex. So really, to get an entire network in the cortex, which it's not the only part of the brain, but it's a very well-studied part of the brain. It's an important part of the brain. The sort of natural scale of a cortical network is a millimeter. It could be maybe half a millimeter along some dimensions, but a millimeter of data, that's a petabyte. And until last year, probably, there have not been any petascale data sets in biology, as far as I know. But this is where we are right now in cortical connectomics. Data collection was hard. We've done it. Segmentation is extremely hard, but we have a first draft of that. And then finally, the informatics, et cetera, to make sense of it, that's going to be the task for the coming years. OK, catch your breath. Now that we have 1 cubic millimeter data set in all of connectomics and some others that are roughly that size, there's a big movement, and I think I'm part of it. I am part of it. Why don't we do something big that will be complete, accurate, and permanent? A data set that can really serve as the foundational resource for answering a huge number of questions about the mouse brain, and that is an EM reconstruction of the mouse brain. I'm not going to talk about the technical hurdles, the techniques, the difficulties, any of it, but I'm just putting it out there, and I'm not alone in this this idea. Jeff Lichtman, Winfried Denk, Sebastian Sung, others, all in the field, are talking about it right now. But it's an exabyte of data, 1,000 petabytes. Let's just sit with that. I'll talk about it later. But I think most of us in neuroscience think about the human brain. Why is it so fascinating? Because the human brain is what makes us us. How many of these tools can be applied to the human brain? And clearly, what's nice about all these numbers is they really fall in powers of 10. It's plus or minus a factor of 2. 1 micron, 1 synapse, 10 microns, the size of a cell body, 100 microns, a local circuit. But a millimeter is sort of the foundational thing for cortical circuits. But then brains, a mouse brain is half a cc, half of a centimeter cubed, and the human brain is a little bit bigger than 10 centimeter cube, a little bit more than a liter. So just add a power of 10, but cube it, and you go from exabyte to zettabyte, 10 to the 21 bytes. Not going to happen. It's certainly, maybe in someone's lifetime in this room, certainly in someone's lifetime in this room, but not going to happen in the next decade. However, we can't do that today, but I've already stipulated, and believe it or not, I think we can work at the exascale. Certainly at the tens or hundreds of petabytes, we can work at that. And there are things that I think will be dramatic and new that we can do at simply a factor of 10 lower resolution, mapping more than half of the axons, of the long-distance axons, the myelinated ones in the human brain. It's an exabyte. The human brain is go back another factor of thousands. It's 1 exabyte at 100 nanometers. And it's merely 100 petabytes at 200 nanometers. And that's a goal that we're setting. And we're not alone in this. Speeding up a lot. Definition of connectomics very quickly is the wiring diagram of a brain or of a brain region. We've heard about that already. Where were we a decade ago? Really the first 2 studies in the field, modern field of connectomics, came out simultaneously in one issue of Nature. In 2011, there was a study on the cerebral cortex, the visual cortex, that combined physiology and anatomy, and sort of wonderfully it was paired with a very similar study, when you zoom out, of function and structure in the retina from Winfried Denk's group. 10 years ago, 8 years ago, the scale of connectomics was hand tracing hundreds, at most a couple thousand connections. Very, very hard work, slightly smaller volumes. For us, recently, 3-4 years ago, we were at the scale of a couple thousand connections. There are similar papers from Jeff Lichtman, a couple thousand connections amongst a handful of neurons. Won't talk about the science there. Sparse tracing by hand of countable numbers of synapses. But recently, we were all challenged to go to the next level by this remarkable program from IARPA called the Microns program, where good money was laid down to study computation in the cortex, but the experimental goal was to do this kind of experiment at the millimeter scale. Do calcium imaging at the millimeter scale, and then EM reconstruction at the millimeter scale. And that's what we've been doing here at the Allen Institute EM Connectomics group, network anatomy group, for the past 4 years. Skipping through this quickly, the goal of the program was to combine neuroscience and data science to understand cortical computation, even to provide inspiration for new types of machine learning. But let me talk about the experiments, and before I do, let's introduce the experimental team, not at the end, but right here. Co-lead, but real lead, Nuna DaCosta, who's sitting right back there, who really runs this group. In the group, there are a number of scientists. I'm only showing the scientists, but there are many other people. Agnes Bador, who has done a lot of things, both in cutting series. All the data I'll show you were hand cut by Agnes, and much of the analysis was done by Agnes. Joanne Buchanan, who's done all of the tissue processing. Forrest Coleman, who who introduced me. Casey Schneider-Mizell. The two of them have been, among other people, doing the analysis of the results Dan Bumberger right here, who sectioned the millimeter cubed data set, or led it with a big team. Wenjing Yin, who runs and is the person who is in charge of the imaging factory we have across the way, and then Shelby Suko, who's our project manager. And it's part of the big consortium with Andreas Telius leading the calcium imaging and computer end, and very importantly, I have his name later on, Jake Reamer, who you've heard from already, doing the calcium imaging, the local team, and then finally Sebastian Sung, who's responsible for the segmentation, his team. This is Jake, who you've already met, in Sebastian's team. In order to get into this game of functional connectomics, the function and connections, you first got to do calcium imaging, which was fun and easy enough for small volumes back in the day, and we do a ton of it here. But it's hard work and amazing to do it for a cubic millimeter. And this is, I think, roughly 70,000 neurons that were studied in vivo with calcium imaging, led by Jake. That tissue goes into the EM pipeline here. In this project, we've had 3 data sets, small one, still quite large, small one called Pinky, one that's a square millimeter by kind of thin, which we call basal, and then the big one, the cubic millimeter, which is mini, all mouse names. The experimental workflow goes from functional imaging, the calcium imaging, through making samples stained with heavy metal, sectioning, which is very hard to begin with, but extraordinarily hard to cut millimeter scale sections times 25,000 order serial sections without significant loss, and certainly without any sequential loss. Imaging was a lot of hard work. That I'll get to a little bit, but just was an extraordinary effort to collect a couple of petabytes of data over the course of roughly 6 months. And then the analysis, the merging of the 2 data sets of calcium imaging and EM, and then finally the segmentation to get connectivity. Here, all the people don't have time. Why was it so hard? I've already done this, factors of 10 things, but just to bring it to life, the experimental challenges, state of the art before this, plus or minus, has been several hundred micron scale. So in the first part of this, we did a volume 200 by 150 by 100 microns, and that's a lot of work. It's 2,000 serial sections, 5 million images, 400-ish neurons, and lots of synapses that we're analyzing. I'll show you a bit of that. But put in the context of a cubic millimeter, it's 1/10 linear, 1,000th volumetric. And again, the collection of the data set was just an extraordinary effort. 150 million images. But the goal of it is to study the connections amongst 100,000 neurons with order 1 billion connections. We do this with microscopes that now it's thinned out a bit. Everyone will kill me. But if someone is really interested in seeing the factory that we have down the way, come to me at the end, and we can send a small group to see it. It's kind of it's a remarkable thing. Again, I think this is emphatically not a talk about technique. It's not really a talk about specific results. So I'm going to rush through how we do it, but it's essentially a standard transmission electron microscope, but tricked out in several ways to take very large, many pixel images and to do it extraordinarily quickly. And then once we do that with a custom stage control, so we can take these very large montages of individual sections, but bringing section after section after section into the microscope because the sections are on a tape. It's almost, the whole thing is almost like a movie projector, but a big one with that sees very small things. And then multiply it by 6 and collect a lot of images. And what do you get at the end of the day? I mean, at the end of the day, you don't just get pictures, and I'll give it a hint of the kind of science that one can do with this, but you've already heard some results, particularly from Jürgen, the kind of results that you can get in this field nowadays. But just the data set itself is, an extraordinary thing. It's on the order of 25,000 serial sections of the top of the cortex to the bottom of cortex pia to layer 6. Huge number of neurons, and for each of these neurons, the entirety of the local dendritic, or of the dendritic arbor, the receiving end, and the local axonal arbor talking to that local circuit. I can keep going. There are black lines that sort of come from the difficulty of this. It's an artifact, but really the success story of the past several years has been the post hoc analysis of data sets like this. They can deal with a certain number of artifacts and can take these raw data that used to be analyzed by hand, and then in an automated fashion segment them. You heard a good deal about proofreading. It's not perfect, and then proofreading has to proceed after that. But to first approximation, certainly for this talk, you get really quite remarkable, sort of way better than one would get with light microscopy in their detail, morphologies of the dendritic arbors, for instance. The dendritic arbors are virtually perfect, and from the dendritic arbors and axonal features, you can do a very good job of classifying different types of neurons. This is an old slide, but in this early volume, there's several hundred excitatory neurons, a number much larger than 10 of inhibitory neurons, and amazing things such as astrocytes, these fractal sponge-like structures that are, when you actually see them, are not your textbook astrocytes. They're extraordinary structures. I don't think this group has seen this fantastic tool called Neuroglancer that Google created for, on the left hand, going through 3D data sets with multiple layers, both of the morphology and of the segmentation, but with the ability also to render meshes from the segmented objects. So I call it one-click Golgi, the standard anatomy of until now, essentially. How do you see neuronal morphology? You sustain 0.1% of the neurons, and you can see that neuron by ignoring all the rest. Now we have every neuron, and pre-computed are the dendritic arbors, and all you need to do is click on it or access it programmatically that I'll get to, and you have the mesh and that you can interact with in 3D, so we can see it and try to learn from seeing it, but also interact in 3D to do all sorts of morphometric analyses, et cetera, et cetera. But this is a wonderful tool that is largely a viewer, but our fantastic analysis team in collaboration with Sebastian Sung's group has made a rich database that interacts with the images, the meshes, and metadata like synapses with a database that really can be queried to ask questions and answer questions very quickly. Again, what are the big questions? Morphology, what are the morphological cell types, but especially using ultrastructural features such as cell biological features such as nuclear morphology, organelle morphology, and location of organelles. Something that's really just starting is the ability to classify neurons based on something that people in cell biology and analyzing light microscopy have been doing for some time, but now we're able to do it with three-dimensional EM data sets for thousands of neurons. Connectomics, what are the determinants of connectivity? I've been through them: topographic, cell type, and functional. And then again, morphoconnectomics, the details that are important for models of the cortex. Can we get synaptic strengths? Can we incorporate geometry into the connection wiring diagram to see how neurons integrate information? Let me unpack what I've already shown you twice, is the ideas of topographic, cell type, and functional specificity. If you're interested in this, this is from a review I wrote a number of years ago, but it's, similar ideas are out there. Again, topographic specificity is, you got to be in the neighborhood to connect. That's simple. Cell type specificity is when you're in the neighborhood, still different cell types connect preferentially to other cell types. I will show you some data on that. And then finally, functional specificity. What about the physiology can tell you how to interpret the connectivity and vice versa? A few slides of results, tools and results from this kind of work from our small data set, just to show the current state of the art. Segmentation is good enough that one can take one of these cells, such as a basket cell here. It's in a type of inhibitory, common type of inhibitory cell in the cerebral cortex. And the dendritic arbor is perfect without proofreading. The axonal arbor is a bit over-segmented, and with an hour or two of proofreading can be made to look like this. And this is, it's an extraordinarily detailed 3D model of a neuron with 4,700 input synapses that we can trace to the sources, and 1,700 axonal boutons, the output synapses. And again, that's not one neuron, that's one among hundreds of neurons in this data set, or tens of thousands of neurons in the new data set. And for each of them, there is this kind of rich segmentation and metadata, thousands of synaptic inputs, hundreds, but soon to be thousands of synaptic outputs, their locations. But that's only the beginning. Let's jump into the question of, let's go cell type by cell type and see what we can learn from this type of reconstruction. Again, very quickly, I'm running through things, but just to give a flavor of where we are now, the group has put together a really lovely story on a classic cell type because of it's so starkly different from other cells. It's called the chandelier cell, an inhibitory neuron that actually does not send its synapses onto dendrites like every other neuron, but sends its synapses to one target, the beginning of the axon of excitatory neurons. And that's one, it's the textbook example of cell type specificity and subspecificity. These neurons send inhibitory synapses onto axons and only the beginning of axons. But it's also, for our small data set, it's a type of, it's a topic that we can really study densely. Even with a small data set, because we have hundreds or more than 100 pyramidal cells, excitatory cells that we have the axon initial segment. And all we need to do to find literally every single one of the synapses from this source is to look at the axon initial segments. And I say literally every single one, not because the literature tells us, but that's what our results have really nailed down. And I was going to show several slides, but I'm just going to show one of this, very rich study, just to give a flavor of it. On panel A is an excitatory cell, and it's big and above the screen. That vertical thing is the beginning of its axon, and in two different colors are two different presynaptic neurons, giving inhibitory synapses onto the axon initial segment. And that's one example, and we have more than 100 examples of those. But what it allows us to do, particularly with the database, is to do this exhaustively. Find every single synapse. In this case, they were found by hand first, but they're also in the database from automated segmentation. And almost the only data, plot I'll discuss in any detail is C, where C, the axes are, the vertical axis is distance from the cell body along this axon, where the top is at, virtually at the cell body, and it goes down to 60 microns. And this is the histogram of all of the inhibitory synapses, which is all the synapses onto all of these 100 some odd axon initial segments. Two types of inputs, chandelier inputs, the textbook, and then other, which are predominantly basket cell inputs that give sporadic inputs. And you can see that not only is there exquisite tropism for a place, but there's also an exquisite tropism of exactly where this red plot is peaked around 20 microns away. So there's just clearly a mechanism to achieve this targeting. And on the right, I won't discuss in detail. It's the numerology, the kind of things that one couldn't do before having an encyclopedic view. How many distinct chandelier cells are there for each target? What's the total number of inputs for each target? How many synapses does each chandelier axon pair make? We can do all of these plots, and now it's just a query. And it's a query of this form, that now the infrastructure that you saw with one-click Golgi, with the human interface, with EM, segmentation, and meshes, there's a fantastic database behind it, and it can be queryable very quickly with simple queries in Python. And so it's transformative in terms of how quickly you can ask questions. This is just a movie to show what it looks like. These are all the inputs to a pyramidal cell in this data set. The inhibitory inputs are blue. Let's look at only the inhibitory inputs and color them. The basket cell inputs, the predominant ones, are on the cell body and sparsely on the dendrites. The chandelier in light blue go where chandelier cells should go, and the yellow ones, somatostatin, go more out here. I don't quite see them. But again, you can see it, but also you can analyze it programmatically. You can quantify it, and finally, here's a summary from a while ago of the types of tropisms we can get. Cell type specific connectivity. On the x-axis, the different stacked histograms, each one of those is a different class of inhibitory axon. And on the left, you see 6 colors. Those are different types of targets. The different subparts of excitatory cells, but also in red, whether inhibitory targets. So where do inhibitory inputs from different classes go on excitatory targets, all the blue things, and the green is excitatory. Did I say excitatory? All of these first 5 are the excitatory targets of inhibitory neurons. And there's some interesting stories there. The only one I'll talk about, just to maybe help you read the plot better, is axoaxonic cells or chandelier cells. It's all of their targets are green. What is green? They're all axons. But here's another interesting thing, and it's sort of stronger than what's in the literature today, and the kind of thing that might, as we do it and as the field does it, yield a number of discoveries, fundamental questions. Inhibitory cells are classified according to the neuropeptides that are seen in them, background information. There are multiple types of calbindin, somatostatin, and VIP. But VIP in the literature is known to target inhibitory cells, and there are several classes of VIP neurons. We think we have found that one of these 2 classes, again, this is work that Nunu da Costa started with Agnes Bador and then has been brought to the larger informatics team. The initial finding is that a certain class of these VIP cells are extraordinarily good at targeting inhibitory cells, that's what they do, unlike every other type, which sort of targets inhibitory cells roughly in the fraction of inhibitory targets that are available, specific connectivity. So in the last few minutes, I'm going to skip this more detailed questions. And in the last few minutes, after I'd really say where I think this is all heading, the more interesting questions in the coming years will be really nailing down cell types, morphological cell types, and their distinct rules of connectivity. And what is one of the biggest stories in neuroscience recently is what are the distinct classes of cell types. And that's just being hammered, particularly by the Allen Institute. This is work from the Allen Institute. Different cells in the cortex can be classified by their transcriptional profiles. And this is just a lovely example of work from the Allen Institute correlating the transcriptional types with morphology. And just the simple idea is one of many ways of going from molecules to wiring diagrams is to start with the molecular signatures, tie that to light microscopic morphology, compare that to EM morphology, and to give the whole picture. There are other ways of doing that. But very quickly, in less than 5 minutes, I will talk about whole brains. I hope I'll do it in less than 5 minutes, but very close. The next grand challenge is the whole mouse brain. There are a number of reasons to do this. I'm just going to list a very small number of them. Why do it? Very simply, along a lot of dimensions I won't discuss, the whole is greater than the sum of its parts. You can do a bunch of circuits, but you don't know how they interrelate unless you do it all together. It'll provide many local circuits, but in the context of long-range connections, what do long-range projections give you? Long-range projections are crucial in terms of subcategorizing different types of cells. For instance, different types of excitatory neurons in the cortex. But perhaps more interestingly and crucially, we might know all the exquisite cell type connectivity within a local circuit, but how the whole thing works is crucially dependent on what kind of targeting there is from one area to another. You can't do that unless you have electron microscopic detail in both places and connect them together. So that's really the big idea. But just in a snippet, if you have the whole thing, the informatics is simpler. It's one unitary data set like the human genome, and it should prove foundational, just sort of stated and to be discussed. now in 3.5 minutes, the human brain. And I've already shown you this slide, or a bit of it. Current state of the art in human brain wiring is diffusion imaging. Why is it not a wiring diagram? Because it simply strings together these vectors and tensors, but really just plain old vectors that you string together and you get notional wires. How much better and there's been a great deal of wonderful science done with this, but why not do more? A single voxel, in many studies, is 1.2 millimeters. This is what, this is actually a sixth of a millimeter, what actual wires look like. They're not going one direction. They're going many different directions. And they have many different sizes and many different sources and targets. But they're remarkably traceable. And it's diverse. At different parts in the white matter, sometimes they're largely parallel with some outliers. Sometimes it's partially structured, and sometimes it's just crazy mixed up. But if you can trace the different, especially these large tubes, which are remarkably easily segmentable, I'll talk about it later, you cannot trace one neuron or 10 neurons, which is what one gets with tracer studies. You can trace billions of neurons in a large brain. The Stark statement is never has a single axon, long-distance axon, been traced from one place to another in the human brain. This technique will, out of the box, trace, if it works, billions of connections. So, it's something you could imagine that one can get passionate about wanting to do this. The background is, it's from antibody staining that's been around for a long time. Axons are very easy to stain with antibodies and trace. The diameters of axons in the human have a range of sizes, but the median size is a micron. So if they're very beautifully stained, and really the size that you can trace is the size of your voxels, of your true resolution. So roughly empirically, it seems that we can trace 1 micron axons with 200 nanometer voxels, and we hope we can get smaller with 100 nanometer. How do we do it? With light sheet microscopy. That's the goal. And here are just a bunch of bullet points. There are 100,000 kilometers of axons in the human brain. Half of them are greater than a micron. You wouldn't want to start this from scratch, but building on the tools from the Connectomics, from segmentation through stitching and aligning, through informatics, it gives a great head start. I just said that. Very importantly, it could serve as ground truth for hundreds and hundreds of studies of diffusion imaging in human brains. Potentially provide new parcellation of cortical and subcortical areas. And here's a real kicker that I truly believe in. Deep brain stimulation works largely by activating axons. How can we not have maps of axons in the human brain? Because it has great clinical importance. And there are a lot of ancillary things. It'll give a complete cell type, cell count, with the wonderful antibodies we heard about the other day. It could do a great deal of getting to cell types. That's a whole other talk about whether we can link these axons to the cell bodies and with other antibodies get cell types, but that's a long-term goal. And then finally, I'm going to end just everything you've seen was in macaque, but in the past several weeks, we've been, one of the great things about the Allen Institute is all the work on human brains. And we have tissue right down there. And this is some work that Emily Tershak, who together with Rylan Larson did the earlier stuff. Emily did staining and imaging of a bit of human cortex and white matter, and it's just fun to see that this works in the human, that the white matter is easily traceable. There are all sorts of diversity of axonal types and directions. Zoom in a little bit, and you can see that, essentially my rule of thumb is that things that are 4, 5, 6, 7 pixels wide appear to be extremely traceable. This is 200 nanometer pixels, but expanded 2x. So now we're at this place, 100 nanometer voxels that would be merely an exabyte if we did the whole human brain. So that's it. I can give a little bit more about what we can learn, but I think I've done that enough. So I just want to say, this is a fabulous place to work. You've seen the people on our smallish team, but it's a very huge group of wonderful people who do work in the mouse and human neurobiology, inspired by our founder, Paul Allen. Questions? Thanks.
Questions for Clay? I think we got one down here in the front.
Hi. Hello? Is this on? Okay. Hi. Thanks for that. So my question is, if you're going to get a 3D EM map of a whole human brain.
Mouse brain, please.
Well, okay.
Well, go ahead.
Let's say it's a human brain.
Okay, let's say that.
Who would it be, and how? What kind of person would they be, ideally?
Yeah, let me repose the question. If you do one of anything, how do you pick the one? And we're talking about doing a whole mouse brain. I am at least talking about doing one human brain with the light microscopic techniques. And of course, the great debate is what fraction, putting off your exact question, what fraction of the discovery will you get for n equals 1? Personally, I think a very large amount. But then, of course, and you pick the way you pick anything, it'll have to be blinded according, sex blind, gender blind, identifiable features blind. We'll have a number of candidates that will be in a blinded fashion chosen to have the best attributes. I think the same will be true of a mouse. But of course, you don't do anything to do one. You do anything because to eventually do many. And the thought of having $1,000 genomes was silly when the Human Genome Project started. But you got to start somewhere. And of course, it will become a tool to study diversity, et cetera, et cetera. That's the simple answer.
Yeah, thank you. That was a great, very informative talk. So do we need to really store exabytes or zettabytes of data, or could you store reduced representation?
Absolutely. It'll have to be. I mean, zettabytes, whatever. Yeah. But exabytes, you could store an exabyte. It would cost a fortune. But this one statement is, what is segmentation but extraordinary compression?
Yeah.
Or better, what are the early stages in segmentation? Extraordinary compression. Of course it'll be compressed. Mm-hmm. And, off the shelf, 3x compressed. But I expect it'll be, 100x compressed. And, exactly what that factor is for EM, I don't know. Mm-hmm. For the light microscopy data, it's far simpler.
Sparse.
I think that the compression will be extraordinary. So, yeah.
And the other question I had was, what's the value of connectivity in fixed tissue versus in a whole fixed tissue versus a circuit, but in a live tissue?
Yeah.
Mapping activity plus connectivity. So if one were to spend resources, which is more useful?
Like all of life and science, you need to do things, but you can't do them in isolation. So, typical question number one when you talk about whole human brains is that question number one. Typical question number two is silent synapses, plasticity, short-term plasticity, et cetera, et cetera. And what's nice is that I think we're almost in the era of electron microscopy as assay. For small volumes, it's cheap enough and fast enough that one can do the experiments that one has to do, which is what are the morphological correlates of synaptic strength, morphological correlates of short-term plasticity characteristics, et cetera, et cetera. And I'm part of a big consortium with Kristen Harris to answer precisely those two questions, synaptic strength and plasticity. How can you learn about that from morphology? So we need both.
A little bit comments on this. I think this is really, really great, enlightening talk with a lot of details, a great review of the history with the great kind of like a progress shows. Okay, so I think this is very nice. But
Thank you.
I kind of like feel that we probably should look at in a different way. That right now, many other people actually look at, generate a really nice survey about Earth, and you are really talking about, try to generate all the data for the solar system, for the Milky Way, for the entire universe. Okay, so that's a tremendous amount of data. That's great exploration. But my greatest concern about this is that I'm not even sure about how to really make this effort sustainable, right? So how to really when you talk about really kind of like get this entire mouse brain and eventually probably many of such data set. Okay, not just one, right? And how to make this effort sustainable? Okay, so yeah.
No, it's a very good point. And we are at a place where we have extraordinary advances, and we're just reaping the benefits of 15 years of work by a decent-sized community to be able to tackle entire local circuits. What next? And one could argue for doing 5 years, a decade of more of same. Similar answer to the previous question. Especially if there are good reasons, which I touched on, for rather than doing 500 small volumes, doing a volume that's 500 times larger. The interactions of them is far greater. Sustainability, I agree. It is not immediately sustainable to do a large number of things that are that hard. But it will happen. It'll get cheaper and cheaper. But I think one reason for doing the big thing first is it provides a scaffolding for what will happen with certainty one, that people will be reconstructing local circuits once, twice, 10 times, a series of 5, parametric in a learning protocol, et cetera, et cetera. And that's a wonderful thing to do, the same way one does modern molecular genomic studies. But it's kind of nice to have a scaffolding to plug it into and have that sort of exist. Whether we need to do that this year or 3 years from now, it's a matter of debate. But that's what I feel.
I think that's all the questions we might have time for now. Thanks so much to Clay for this fantastic wrap-up.